Here we go. So, as I was just saying, I want to flap my gums about sympatric speciation. We've talked about allopatric speciation, where you have an ancestral population that's divided by a barrier in space. That's either the organisms stay where they are and a physical barrier arises that splits the range in two, and that's vicariance. Alternately, you can have members of one population dispersing to a new habitat, crossing a barrier that they do not normally get across. If the barrier is the sort of thing that the organisms can cross very easily, then it isn't really allopatric speciation. It's just a population expanding its range. But if a few members of that population found a new population on the other side of a barrier that they don't routinely get over, that would be dispersal. And then once you have these separate populations, genetic drift, natural selection, mutation, and a few odd wrinkles like the founder effect will tend to make them become increasingly different from each other until at some point they're no longer able to interbreed, or at least can't interbreed often, even if they do come back into contact. Peripatric speciation, by the way, is a form of allopatric. It's not really conceptually different, but peri, of course, means around, as in perimeter. Peripatric means around the homeland, and it just refers to the fact that smaller populations may find themselves isolated right on the fringe of the ancestral species range. If you have a lake, and thanks to a quirk in the climate change, the lake dries up just a little bit and the water level lowers, you might have some small ponds that are kind of left behind around the fringe of the bigger lake. And it's in those small ponds that you might well get speciation happening. So peripatric's not really different from allopatric. It just refers to cases where those isolated populations tend to pop up on the margins of the range of a larger population. Sympatric speciation happens without a physical barrier. Remember, sim or sin means together, and patria means your homeland. So this is speciation without a barrier. There's many cases known, mostly in the plants, but not entirely unheard of among the animals, that are due to chromosomal causes, specifically through polyploidy and through hybridization. Hybridization's neat. It is possible to cross two species and get a third. And in fact, you'll see some documentation of that coming up. Those can happen extremely fast. They might take only one generation to act. There was debate that was still going on when I was in grad school back in the lower Pliocene as to whether sympatric speciation could happen gradually. And it now seems that it's not only possible, it's more common than we previously suspected. The orthodoxy back in the days of grunge, when I was in grad school, was that allopatric speciation was by far the most common, but there's a lot more cases known of sympatric speciation. So we'll look at that first, hybridization and polyploidy. You can get two plant species that will hybridize, and the hybrids that you get may not be like either parent. And what's more, that new hybrid may not be interfertile with either of the parents. So remember, when you cross domestic cats and servals, the F1 hybrids were bred back to one of the parental species, the domestic cat, So now we have this breed of savanna cat that is something like 1/8 to 1/16 serval and 7/8 or 15/16 domestic cat. So those F1 hybrids could backcross to the domestic cat parents. But sometimes you'll get a hybrid that's not cross-fertile with either parental species. If there's only one of them in animals, that could mean that the hybrid's not going to go anywhere, that it won't produce anything new. But remember, plants have a remarkable ability to, in some cases, self-fertilize. So even one hybrid can sometimes establish itself as a new population that doesn't interbreed with either parental species. And you'll see a nice example of that in a bit. And then polyploidy happens because of errors in meiosis. I've read somewhere that something like 1 in 300 human conceptions is actually triploid, because either the egg or the sperm was diploid, carrying 2 of each chromosome instead of 1. Those are almost never born, incidentally. Triploid embryos virtually never come to term, but it does happen. And because of mistakes in meiosis, polyploid races of plants may arise, and they may not be cross-fertile with either parental species. And when that happens, all we can say is that, hey, we've got a new species of plant because it can't backcross with its parents. And also it often looks different. And this is very common in plants. An estimated 15% of flowering plant species are thought to have arisen by relatively recent polyploidy, together with 31% of the ferns. And you can see this very quickly if you look at cases where closely related species in the same genus turn out to have multiples, chromosome numbers that are small whole number multiples of each other. So there is Chrysanthemum sigetum, 2n equals 18. I think Chrysanthemum sigetum has leaves that are actually edible and used in Japanese cooking. I think that's the one. There's Chrysanthemum coronarium. Looks very much like sigetum. They're clearly both chrysanthemums, but 2n is equal to 36. And then the one that I think is most commonly sold in your average garden stores around this time of year is Chrysanthemum indicum with 2n equal to 54. So here you've got 3 plants in the same genus, and their chromosomes are either 1 times 18, 2 times 18, or 3 times 18. The simplest way to explain that is simply polyploidy, that Chrysanthemum coronarium could have arisen if you had a diploid egg and a diploid sperm of sigetum finding each other and fusing and giving rise to a zygote. Just here's another example. These are a couple of flowers in genus Anemone, Rivularis, n is equal to 16, Quinquifolia, n is equal to 32, and I include these pictures of the chromosomes so you can see that it's not only that the chromosome numbers are doubled, but the chromosome types are doubled. Like Rivularis over here has, those are 6 acrocentric chromosomes, which means that the centromere is over towards one of the ends. This one has 12 acrocentric chromosomes. So it's not just that they've got double the chromosome number, it's double the types of each chromosome. You could I don't have that data, but you could go farther and look at banding patterns, and you probably see that quinquefolia's set of chromosomes is basically a double set of ribularis chromosomes. For some reason, and I have not really studied this, polyploidy does not always seem to cause speciation. This is a flower that's up right now. I've seen lots of it on, yards in some of the older neighborhoods of Conway. There's probably some coming up just outside Lewis, anywhere they haven't been too quick to mow. You'll see Claytonia virginica about this time of year. This is called spring beauty. And it's interesting, they range in color from almost pure white to some with very strong pink. And Claytonia virginica comes in 3 diploid, quote, races, with 2n equal to 12, 14, or 16. Plus, you can find polyploids of all of these going up all the way to 2n equals 191. Yes, 2n is an odd number. For some reason, that doesn't seem to form barriers towards interbreeding. It's possible that it does. I'm not aware of carefully controlled studies on interbreeding different chromosomal races of Claytonia with each other and seeing if you get fertile offspring. That would be interesting for somebody else to look at one of these days. But from everything I've been able to find out, plants with different ploidy levels can still interbreed, and I don't really understand why. Dr. Noyes would be a good person to ask about this. I've asked him, and he's not really sure either, but one of the things he does is he works on, the evolution of plant chromosome numbers, and plants are basically just perverts. Plants are just weird. Plants do all sorts of crazy chromosomal things that you wouldn't catch any animals doing, especially not in this state. Right. 1928, Soviet scientist Karpachenko gets idea to cross radish and cabbage. Both of these are in the same family. They're both mustards. So they're both in the Brassicaceae. Same family as cabbage, turnips, rutabagas, canola, the plant whose seeds are crushed to make cooking oil, canola. What else is in there? Mustards and Brussels sprouts and things like that, and of course radishes. Now, they've both got the same number of chromosomes, 2n equals 18, and you can crossbreed them and you'll get F1 hybrids, but the chromosomes can't pair up properly at meiosis, so the F1 hybrids can't make eggs or sperm. Remember that at meiosis, the chromosomes have to be able to pair up and do that crossing over thing that they do. And for some reason, radish chromosomes and cabbage chromosomes cannot pair up with each other. So normally you get infertile F1 hybrids. Very rarely, however, an F1 hybrid will produce diploid eggs. So each egg has 9 chromosomes from the radish and 9 chromosomes from the cabbage. And very rarely an F1 hybrid will also produce diploid sperm, each of which has 9 chromosomes from the radish and 9 chromosomes from the cabbage. And if you can get a diploid egg and a diploid sperm together, you'll end up with a zygote that has 18 radish chromosomes and 18 cabbage chromosomes. This is called allotetraploidy. Tetraploid means 4 copies of each chromosome. Allo means other. So you have 4 copies of each chromosome. 2 came from the radish, 2 came from the cabbage, and you end up with a plant that is perfectly fertile because when it produces its own gametes now, every radish chromosome has another radish chromosome to pair up with, and every cabbage chromosome has another cabbage chromosome. So you basically have two of each radish chromosome and two of each cabbage chromosome in every cell. So you end up with a plant with 36 chromosomes. Now, Karpechenko was trying to breed super plant that have delicious leaves like cabbage for cabbage soup and delicious roots like radish. Unfortunately, natura dura, sudba indieka i жизнь копейка. Sorry, that's a Russian proverb for nature is a fool. Fate is a turkey and life's not worth a penny. What he got was not a plant with the cabbage leaves and the radish root. He got a plant with the cabbage root and the radish leaves. This did not save Soviet agriculture and is possibly why Karpechenko ended up in Gulag. So he got this plant that he called Raphanobrassica. Actually, these days, for some reason, it's often called Brassica raffinus. And it looks like this. It is not the super vegetable that Karpechenko was hoping for. This is what it looks like. It does have some use as a fodder crop. This is the kind of thing you can chop down and let ferment to make silage and feed it to your cows if you're into feeding cows. But yeah, Raphanobrassica really did not go anywhere. In 1982, the same approach was used to develop hybrids between radish and Brassica rapa, which is turnips, also bok choy and mizuna. That's a Japanese green. So that's an allotetraploid, a plant that has, where each cell has two copies of the chromosomes from each one of its parents. Incidentally, there's some, before I hit the sunflowers, there's some examples of this sort of thing happening in the wild. One example is European and American seagrasses in the genus Spartina, for example. Used to be found on separate continents. I think the American seagrass got introduced to Europe probably through sailing ships. That's a common way for critters from one ocean to be introduced into another. It still is. And by about 1900, there was a new species of seagrass that was found in Europe that actually outcompetes the old one, Spartina anglica, and it seems to be a naturally occurring allotetraploid of American and European seagrasses. So there are cases of this happening in nature that we've documented. Okay, a little more complicated. Let's look at sunflowers. There's the annual sunflower, Helianthus annuus, from Western North America. This one, I think, is the ancestor of our domestic sunflowers. And Helianthus petiolaris is the prairie sunflower. There's a third rare wild species of sunflower called Helianthus anomalus that was already thought to have arisen from crosses between annuus and petiolaris. The two live in the same range or overlapping ranges. It would be pretty easy for pollen of one to come into contact with egg cells of the other. And so we already think there's a natural hybrid of them called Helianthus anomalus. Anomalus is ecologically specialized. The parents are generalists that live on the prairie. Anomalus lives in sand dunes. And there are several differences from the parental species through things like chromosome inversions and fusions. Now, Annuus and Petiolaris almost never interbreed in the wild. But if you grow them together and hybridize them in the lab, now and again, it does work. And lab-produced hybrid lines can be propagated. You can make them hybridize and raise the hybrids, and the hybrids will reproduce with themselves. So you can end up with a hybrid population in the lab that's stable. And Loren Riesberg did this, and in 2000, he published the results of some experiments that he'd done, and they look like this. What this diagram shows is the percentage of fertile seed produced in crosses. So he created 3 different hybrid lineages by crossing Annuus and Petiolaris and then getting the hybrids to reproduce with themselves. He did that 3 times and found that all 3 of these hybrids were interfertile with each other, with about over 70% of the seeds were fertile when he crossed his hybrid lineages 1, 2, and 3 with each other. When he crossed his hybrids with Anomalous, the species that we already think is a naturally occurring, or the descendant of a naturally occurring hybrid, you can see those lines are thick. This hybrid crossbred with Anomalous, and about 37% of the seeds were fertile, 40, 42. So these hybrids that he produced are at least partially interfertile with Anomalous. But none of the hybrids were nearly as fertile with either one of the parents. All 3 of the hybrids, here's a 20, a 14, and 8.6. So only between 8.6% and 20% of the seeds were fertile when he crossed his hybrids with Annuus. And then when he tried to cross them with Petiolaris, 0.84%, 1.09%, 1.29%, roughly 1% of the seeds were fertile when he tried to cross his hybrid lineages with Helianthus petiolaris, one of their parents. And then when Annuus and Petiolaris were crossed with Anomalous, they're also almost completely infertile. Fewer than 0.2% of the seeds were fertile when Annuus or Petiolaris was crossed with Anomalous down here. So here's what we're getting at. He was able to create these hybrids in lab. They're fertile with each other. They're partially fertile with this wild species that we already thought was a hybrid, Anomalous. His hybrids are not very fertile with either one of the parent lineages. So what he'd basically done is recreated a speciation event in the lab. He'd basically replicated the wild cross that we think gave rise to Helianthus anomalus, and showed that he got hybrids that were fertile with each other and partially fertile with Anomalous, but not very fertile with their parents, just as Anomalous is not very fertile with its parents. Okay, so that's a case where Riesberg was able to duplicate a speciation event happening in the wild. The weird thing is that the same thing appears to have happened 2 more times. This is Helianthus deserticola that lives on desert floors. Helianthus paradoxus lives in desert salt marshes, and you might remember that that first hybrid species we talked about, Helianthus anomalus, lives in dunes. All 3 of those are pretty extreme environments. Those are all tough places for plants to make a living. They all inhabit niches that their parents can't live in, and they don't do well in their parent species niches. These don't grow very well on prairies. The idea is that possibly when you hybridize species like this, if you create new species through hybridization, maybe the combination of genes that you get when you do some of these hybrids, you get new combinations of genes, and that might actually be advantageous in colonizing difficult environments. You know, maybe hybrids like this that have genetic material from different species are somehow better suited, better, yeah, more capable of colonizing difficult habitats because maybe they have, you know, a greater range of genetic resources to draw on. Anyway, that's the thinking. Riesberg, last I checked, had not really followed up with a lot of this. He was working on other species. So I'm not sure what the current thinking is here, but it seems to make sense, at least to me. So again, I mentioned earlier that 15% of angiosperms were recent polyploids or hybrids. If you look at more distant history, it goes up to, you know, estimated 70% of flowering plants would be derived from polyploids or hybrids if you go farther back in time. And again, we think that allows plants to colonize extreme habitats because it generates new traits, new combinations of traits. And it's also been extremely important in the development of crop plants. Here, this is cool. This is something that was actually worked out before the the Second World War by a Korean scientist working in Japan. And his name, I believe, was Dr. Yu in Korean. We'd probably write that as Y-O-O, I think. But in Japanese, which he had to publish in, his name is just represented as the letter U. So this is the triangle of U. I think it said the triangle of U. And by looking at karyotypes of different plants, he worked out that Brassica rapa, which is the wild Brassica species that gave rise to the turnip, crossed with Brassica oleracea, the cabbage, produced Brassica napus. Napus is rutabagas. Don't know if you've ever had those. They're these huge kind of misshapen roots that you can see, you know, the grocery stores around here carry them. Brassica napus, by the way, is also grown for its seeds, which are crushed to yield canola, one type of vegetable oil. Brassica rapa crossed with Brassica nigra, that's black mustard, gives you Brassica juncea, which is yellow mustard and edible greens. Yellow mustard is the seeds crushed into a paste together with vinegar or white wine or, you know, other spices or whatever it is you want. That's the stuff you put on your hot dog. It's a paste of Brassica juncea seeds together with some liquid and other flavorings. Brassica juncea also gives you edible mustard greens. And incidentally, you make it a triangle. If you cross oleracea with Brassica nigra, you get a species called Brassica carinata, known as Ethiopian mustard. It's not something that I think we get a lot of here, but it is cultivated in certain parts of the world. So you have three ancestral species, Brassica rapa, oleracea, and nigra. Each one of those has hybridized with the other two to give you three more edible species, Brassica napus, Brassica juncea, and Brassica carinata. Similar things have been done with bananas. Now, back in the day, hi, sweetie. Morning. Back in the day, I got a brochure once. I got a flyer from somebody who was preaching that old time creationist religion. And it was called The Banana, the Atheist's Nightmare. And it gave these 10 reasons why bananas clearly showed intelligent design, not, evolution by random chance. The banana was clearly designed for humans to enjoy. It had a perforated wrapper with a tab for easy peeling. The wrapper was biodegradable. The taste was pleasant. My favorite was that the banana curves towards your face for ease of eating, which surprised me because the banana's always curved away from my face. I could never figure out what I was doing wrong. Anyway, here's the thing. I used to have an actual wild banana plant growing in front of my my apartment in New Orleans. And I actually tried eating one of the bananas that it grew. It grew just fine. It produced bunches of bananas, and I tried eating one. The peel was about 2 inches thick. I had to cut it with a knife because I couldn't peel it at all. The flesh tasted like soap, and I almost chipped a tooth on the seeds. Wild bananas are loaded with seeds. They also don't taste that great. And the reason for that is that cultivated species of bananas are triploids. They don't produce any seeds because they're triploid. They've got 3 of each chromosome. So when the chromosomes pair up in meiosis, they can't, because you can't pair up a triplet. And that's why seeds are not produced. And I believe that's the same way that you get things like seedless watermelons and seedless oranges and things like that. They're triploids. And depending on the variety of banana, they're either autotriploids of a species called Musa acuminata, or they're allotriploids between acuminata and balbiziana. And I think you have some varieties that have 2 acuminata and 1 balbiziana chromosome set, and some that are 1 acuminata and 2 balbiziana set. Okay. One moment, folks. Sorry, cat knocked over a glass of water. It's okay. Yeah, I tried to salvage what I could, but I don't think I got everything. Sorry about that. It'll be fine. Okay. All right. So that's why cultivated bananas don't have any seeds. That's why they have to be propagated asexually by cuttings. They can only be propagated by human intervention. And in fact, they do show intelligent design, but it's design by human breeders. Bananas have all of those features like a pleasant taste and a perforated biodegradable wrapper because humans have made them like that. The same is true for apples and other fruits. I think apples are actually hexaploid, if memory serves. You can't grow an apple. And that's the reason why, if you bought a grocery store apple and you planted the seeds, it might grow, but you would not get anything remotely like the same variety of apple that you got the seeds from. They don't breed true. Yeah, their chromosomes are just weird. Ooh, cultivated wheat's a great example. Mm-hmm. Yeah. Oh yeah, my wife reminds me that there was a real guy named Johnny Appleseed who traveled through what was then the Northwest. It's now like Michigan, planting apple trees everywhere. And he got his apple trees by growing apple seeds, hence the name Johnny Appleseed. But if you grow apples from seed, you usually don't get edible apples, but you do get very sour apples called spitters that don't taste great, but they're terrific for making booze. Johnny Appleseed was raising his apple trees not to make delicious apples, but to make hard cider. Anyway, back around, about 10,000-ish years ago, in an area called the Fertile Crescent of what's now Iraq and Turkey, the local hunter-gatherers started collecting seeds of wild grasses one in particular called Triticum urartu. And they figured out that if you took the seeds and you threw them down on some unoccupied ground, they'd grow and you'd get more Triticum urartu. So this is one of the species that was developed, and it's produced cultivated varieties that are called emmer. Emmer is actually still grown in a few parts of the world. It's not all that productive, but it can grow in dry conditions and poor soils. So I think there are parts of Italy that still make emmer bread, for example. Well, emmer hybridized with a grass that was probably a weed in the fields called Aegilops speltoides. And when it hybridized, they produced a new variety that had chromosomes from both emmer and Aegilops called Triticum turgidum, known as durum wheat. Durum is still grown. They grow a lot of it up in places like North Dakota. It's very high in protein, and that makes it the best for making pasta. If you have, anytime you've had spaghetti noodles or anything like that, it was probably ultimately made from durum wheat. But when durum hybridized with another weed in the fields called Triticum tauschii, it produced bread wheat, Triticum aestivum, which is the variety used to make most wheat flour and thus most baked goods, bread and cakes and things like that, ultimately come from Triticum aestivum. And Triticum aestivum is an allohexaploid, meaning 2n equals 42. 14 of those chromosomes came from emmer, and 14 of its chromosomes came from Aegilops speltoides, and 14 of its chromosomes came from Triticum tauschii. So it's an allohexaploid. And you can even take it further, because bread wheat has been crossed with rye to form an allooctoploid plant. 14 chromosomes from emmer, 14 chromosomes from Aegilops, 14 chromosomes from Triticum tauschii, and 14 chromosomes from rye, genus Secale, to form a plant called Triticale, which I'm not entirely sure what it's used for, but I think I've seen Triticale on the ingredients lists of some of the higher end cereals that you can get at Whole Paycheck, or possibly at Bougie Kroger over on Salem. Some of those higher end, organic cereals, I think, have some Triticale in them. So we've developed quite a number of crop plants, not always knowing what we were doing. I think I mentioned that bread wheat may have come about from different wheat varieties hybridizing with random weeds in the field. But whether we were conscious of it or not, we've created a number of important food crops through hybridization and through polyploidy, taking advantage of these things that plants do. It's not nearly as common in animals for reasons that I don't fully understand. And I've talked with Dr. Noyes about it, and he's not too clear on it either. This seems to be kind of a botanical mystery. Plants can tolerate a lot more screwing around with chromosomes than animals can. So very often, if we have extra chromosomes or extra chromosome sets, or chromosomes from different species blending together in animal bodies, the result is often sterile or defective offspring. But there are some neat cases where we do have these happening, and a classic one is whiptail lizards in the American Southwest. The New Mexico whiptail lizard is a cross between the western whiptail, Cnemidophorus tigris, and the little striped whiptail, Cnemidophorus inornatus. If you hybridize these two, you get a third form, Cnemidophorus neomexicanus. The weird thing is that Cnemidophorus neomexicanus is all female, and it cannot interbreed with either one of its parents. And the reason is it's parthenogenetic, meaning that it can essentially produce fertile eggs without the need of sperm at all. There's various ways in which different organisms can pull that trick off. In some cases, you might know that when eggs are produced through meiosis, instead of 4 eggs, you get 1 egg and a couple of tiny lumps called polar bodies. Sometimes one of the polar bodies can just circle right back and fertilize the egg the way that a sperm would. Other times it's different. I'm not really up on all of the mechanisms how this can happen, but a number of organisms do carry out parthenogenesis, meaning that they produce fertile eggs without the need of a partner. And Nematophorus neomexicanus is an obligately parthenogenetic species. This has been observed in some other lizards, certain geckos, for example. Okay. So there's Nematophorus tigris and Nematophorus inornatus right there. When they interbreed, what you end up with is Nematophorus neomexicanus, which is all female and parthenogenetic. So there. Can we get sympatric speciation happening gradually without some of these chromosomal tricks? Can you get sympatric speciation happening on a gradual time scale instead of just within a couple of generations? Up until about the time I entered grad school, I guess that's not a few years anymore. I'm not that old. The stock answer was no. What I was taught was that allopatric speciation was the way to go. And if sympatric speciation happened, it was rare. Here's one of the first cases that was documented. Ragoletus pomonella, known as the apple maggot fly. If you find a worm in your apple, it's probably not like an earthworm, because earthworms live in soil and don't eat apples. It's probably a maggot. And Ragoletus pomonella is an agricultural pest in many areas. The adults lay their eggs on fruit, and the larvae burrow into the fruit and feed on it. Here you can see a couple of maggots in this apple. After the fruit falls to the ground, the larvae burrow out of the fruit and crawl into the soil, and they pupate there. By the way, if you ever decide to go into the organic fruit business, fruit is one of the most pesticide-heavy crops that we produce, partly because we've all been kind of grown up with fruit that looks absolutely perfect, that, you know, not only is delicious and all that, but doesn't have any blemishes at all. You know, fruit that looks like it might as well have been made out of wax. All right. Okay, right. Sorry, my wife points out that though these days you can buy ugly fruit, and what rejected produce is kind of becoming a trend. Anyway, there are an awful lot of fruit pests that if you're a commercial fruit grower, you have to spray for. Some of them are pretty awful. You know, I've heard of migrant workers accidentally getting sprayed with what they have to put on strawberries and ending up with permanent neurological damage. But if you ever want to grow organic or less pesticide fruit, you can break the life cycle of Ragoletus pomonella just by cleaning up the fallen fruit and not letting it stay on the ground. Because if you let it stay on the ground, then any Ragoletus maggots that are in the fruit can crawl into the soil, pupate, and come back next year. But by cleaning up your fallen fruit, they can't do that. Okay, that was probably more than you needed to know. So anyway, the adults will emerge in summer, fly around, do their thing. lay their eggs on apples, and it's the circle of life, and it moves us all. It turns out that although it's called the apple maggot fly, Ragoletus pulmonella can actually lay its eggs on a variety of fruits. In particular, there's two main varieties of pulmonella that are established in the north central United States. We call these the apple race and the hawthorn race. And some studies that came out in the late 80s, early 90s focused on Ragoletus pulmonella apple and hawthorn varieties in northern Michigan. And here's what they found. The open curves give you the percentage of flies that emerge, and you can see the apple race. Let me go over here to the, better yet, a highlighter right here. Those are the apple flies, and they begin emerging in mid-June. The peak of their emergence comes in mid-July, roughly July 20th or so, and then falls off pretty rapidly after that, and they've pretty much all emerged by the end of August. So this is when the apple flies emerge. The flies on hawthorn begin emerging in early August. They reach their peak in mid-September, around September 15th, and then fall off pretty quickly after that, and they've pretty much all emerged by the middle of October. So the apple flies and the hawthorn flies are not separated in space. You could find them maybe even in the same field, but they're separated in time. The apple flies come out earlier, and in both cases, the timing is synchronized to the fruit that they're growing on. You can see that apples, that pen color, apples start coming ripe in northern Michigan towards the end of July and reach their peak around the beginning of September. You can see there's roughly a 6-week gap right here between the emergence of the apple flies and the peak of the apples. They track each other almost perfectly. Whereas the hawthorn flies, hawthorns start becoming ripe in early September, and they reach their peak here towards the end of October and then rapidly fall off. And the last ones of those might ripen in early November. And again, that's an offset of roughly 6 weeks between the flies emerging and the fruit emerging. So it takes roughly 6 weeks for those flies to emerge and become sexually mature and start laying their eggs. Okay, so you're seeing what I'm getting at. We have populations that are not separated from each other in space. They could be next door neighbors, but they've come to be separated from each other in time. The apple flies mature earlier so that their adults will reach sexual maturity at the same time that the apples on the trees are coming ripe. And the same for the hawthorn race. They reach sexual maturity later in sync with when hawthorns are ripening. So we know that they can hybridize, but hybridization is very low. Allele frequencies are completely different at all loci that have been studied. So these are populations that are not exchanging very many members. And hybrids would presumably suffer reduced fitness because a hybrid might be born in between at a time when the apples are mostly gone and the hawthorns aren't ready yet. So we think there would be selection against hybrids because they're not timed. They're not synchronized to lay eggs either with the apples or with the hawthorns. We actually know that the apple flies aren't perfectly adapted to apples. They'll survive better if you rear them on hawthorn. Maybe apples are not quite as nutritious. But on the other hand, because you can burrow more deeply into an apple than you can in a hawthorn, apples provide better protection from parasitoid wasps that try to lay their eggs in the maggots. So less good quality food on an apple, but better protection from parasites. And the punchline that I didn't tell you guys is that apples were not present in North America until Europeans introduced them. People like Johnny Appleseed, who came through Michigan, planting his apple trees and selling them to settlers, and walking around with a tin pot on his head and preaching this weird religion and selling people apple trees so they could make hard cider. Yeah, the real Johnny Appleseed was kind of different from the Disney version. So we know that this process of speciation must have begun only in about the past 200 years, because there haven't been apples in North America, certainly not in Michigan, where the study was done, until roughly 200 odd years ago. So any changes from the hawthorn fly, which is native, to the apple variety of the fly on this introduced species of apple, any of that change must have taken place only within 200 years. And within 200 years, we have a case of incipient speciation, two populations that are maybe not quite separate species because they can interbreed, but mostly separate species on their way to becoming separate species because they're adapted to host plants that are separate species that are separated in time, in when they blossom and when they produce fruit. So we think that speciation can happen sympatrically if populations become ecologically isolated, adapting to different habitats. This may explain the origins of some other wild species like blueberry maggot flies, for example, Ragoletus mendax. Another case of this was done, studied by a gent named Dolph Schluter in the 90s. This is when this was documented. There's our old friend, the sticklebacks again. And I introduced them back when we were talking about sexual selection, because the females like males with red bellies. And I mentioned that dad's degree of red on his belly was correlated with offspring resistance to parasite loads. Now, sticklebacks can live in the ocean or on lakes. And there's a lot of stickleback populations in the lakes of British Columbia on the Pacific coast of North America. And we happen to know those lakes did not exist 12,000 years ago because that part of the world was covered in glaciers. You may have heard of the glaciers, populated by depressed mammoths and psychotic saber-tooths and really stupid ground sloths named Sid, and these weird saber-toothed squirrel things chasing after nuts. Right? You've seen Ice Age, I assume. Yeah, that. Anyway, so sticklebacks didn't swim from the ocean up into these lakes until after about 12,000 years ago, was when those lakes had to be colonized. Before that, the lakes didn't exist. There were just glaciers. They would have been completely frozen. And it turns out that if you look at the lakes, you have populations of sticklebacks that are limnetic, which means they live high in the water column, and sticklebacks that are benthic, which means that they live down on the bottom. So up there at the top, there's a male limnetic stickleback from one of these lakes. You actually get these doublets of limnetic and benthic populations in several different lakes. You can see the limnetic is more lightly built. He's got more spines. There's a slightly more upturned mouth because the limnetic is probably picking insects off the water surface. The benthic stickleback you can see is chunkier, he's heavier, he's not quite as spiny, he's not as stripy. He's got, different proportions. You can see those cheekbones are a different shape. So male limnetic and male benthic sticklebacks look different from each other. There's no physical barrier between the top of a lake and the bottom of a lake. I mean, it's all water. Benthics and limnetics are capable of swimming into each other's habitats, I suppose. In the lab, you can make them interbreed, and the hybrids are perfectly fertile. So you can hybridize benthics and limnetics, but they're behaviorally isolated because the courtship behaviors have become so different. In fact, if you put a male benthic and a female limnetic together in the same tank, the male benthic will attack the female limnetic instead of trying to breed with her. They no longer recognize each other as potential mates. We know that the strains look different. They feed on different foods. They select different nesting sites. So again, they're behaviorally isolated. They're ecologically isolated. They can hybridize, so maybe they're not really biologically separate species, but hybridization seems to be rare. There seems to be selection against the hybrids, that's speciation by reinforcement. So again, we seem to have two different entities that are mostly kind of separate species, but not quite, but mostly. And remember that in a world where evolution is happening, we should get a lot of cases of this. And this is what I call the factor. Remember that. The principle of will stand you in good stead in many fields of science. Yes. One last case I'll hit you with. This was a pretty recent study of populations of European flounder in the Baltic Sea. It used to be called Platychthes flessus. I take it you've heard of flounder. These are the ones where as they grow, they start flopping over onto, in this case, the left side, and the left eye migrates over to the right side, and they live pretty much permanently lying on their left side with both eyes on the same side of their skull looking upwards. They're also quite delicious, I might add. And here's the Baltic Sea. If you don't know your geography, there's Norway and Sweden up there, Finland on the other side of the Gulf of Bothnia. Russia is off to the, well, of course, Russia used to be on the far left, but they're now on the far right. Never mind, a little political humor there. And the thing about the Baltic is it is connected with the wider world ocean, but only through a fairly narrow passage. Right over here is a fairly narrow strait where Baltic water does eventually mingle with the North Atlantic. That narrow passage, by the way, is called the Kattegat. And yes, that's where they got the name for Ragnar's kingdom in the TV show Vikings. Although in the real world, the Kattegat is not an area of land at all. It's these straits in the ocean that connect the Baltic with the North Atlantic. But because the Baltic's only got a narrow connection with the North Atlantic, Baltic seawater takes its sweet time mingling with the water of the world ocean. And you get several cases of this where you'll have bays or inlets like the Red Sea or the Gulf of California, where they're only connected through a fairly narrow connection with the wider ocean. And so the water that builds up in them doesn't freely and frequently mingle with the water of the rest of the ocean, and it can develop its own characters. It can become more salty, or in this case, less salty. There's lots of rivers flowing into the Baltic. There's a lot of fresh water coming into the Baltic, especially in spring when the winter snows are melting. and it ranges along its length way up here, up in northern Sweden, or over here, which is where St. Petersburg is. It's almost fresh. The scale up here, you're seeing salinity in parts per thousand. 0 would be perfectly fresh. Over here at the Kattegat, it's 20 parts per thousand. Average ocean salinity is 35 parts per thousand. So this is brackish water, not nearly as saline as the global ocean average. And it ranges from still pretty salty over here by Denmark to almost fresh over here in places like St. Petersburg. Why does this matter? Flounder eggs will float in salt water, but they'll sink in fresh water. So flounder that are living in the saltier areas produce eggs that float in the open sea. And that's called pelagic spawning, by the way, where the eggs float as they develop. In the fresher parts of the Baltic, the eggs stay down on the bottom, and they stay close to the coast, and we call this demersal. Demersal, okay, a benthic organism is one that lives all of its life on the bottom. A demersal organism is one that can swim, but spends most of its time down on the bottom. So a demersal fish species can swim up if it needs to, but it's going to spend most of its time lurking down on the bottom. And demersal spawning refers to eggs that develop sitting on the bottom in the fresher parts of the Baltic. So yeah, this is what flounder eggs look like as they develop, by the way. And so you have populations of pelagic spawning flounder and populations of demersal spawning flounders. And it turns out they almost never interbreed in the wild. What you're looking at here is they've taken the frequencies of a whole bunch of alleles and use those to generate some numbers called principal components. And you can think of a principal component as taking a lot of statistical variation in a lot of different axes and boiling it down into a single number or a manageable, relatively small number of numbers. And the demersals are all over here. There's a few demersals that cluster with the pelagics over here, but there's no pelagics over in the demersal cluster. And this is just a way of saying that demersals and pelagics have very different allele frequencies at many loci, meaning there's virtually no gene flow between them. You see the same thing over here, showing you the same thing. I assume this is for a different set of genes. There's evidence been presented for selection on demersal flounder genes that are connected with adaptation to salinity, adaptation to lower salinity, by the way. And so just a few years ago, because it turns out the demersal and the pelagic spawning flounders are not interbreeding with each other, the demersal flounders have been named a separate species. They're now called Platychthes solemndali. They don't interbreed. The demersal flounders are showing evidence that their natural selection has actually acted to make them more tolerant to fresh water. And the punchline is that the Baltic Sea only opened up to the world ocean about 8,500 years ago. When the glaciers were covering northern Europe, the Baltic didn't exist. Well, it existed, but it was this valley gouged out by glaciers and trapped under something like a mile-thick ice sheet. When the glaciers retreated, the Baltic Sea was a freshwater lake for a while. And it did not permanently open up to the world ocean until about 8,500 years ago. So that speciation between demersal and pelagic flounders has only happened in the past 8,500 years. That's one of the fastest cases of speciation ever recorded in a vertebrate. And that's the time scale of speciation. It tends not to happen on, okay, if we were studying the fossil record of fish, 8,500 years is absolutely nothing. If we were studying living fish, 8,500 years is thousands of times longer than any funding agency is going to give you a grant to study them. So speciation can be tricky to observe because it's so fast when you compare it to the fossil record. You know, seeing speciation in the fossil record is often like trying to time a track meet with a calendar, right? The fossil record is just a much more low resolution instrument for measuring time. But speciation often happens under a much longer time scale than a human lifespan. But this is one case where we can time it pretty well because we know it has to have happened in the last 8,500 years, because before then there was no Baltic Sea. And I will have more to say on Thursday about timing and tempo of speciation, and we can talk about Stephen Jay Gould and Punk Eek and all that. And I'll go ahead and stop the recording now.